Okay, so this is the first section of Option A, Neurology and Behavior. Option A has 6 sections. I don't know if we're going to do 6 sections. I might skip around in it. Depends on how much time you're going to be in here this year. So, but we are going to take a look first at the development of an organism, including you, because when you're all starting out, it's all pretty much the same thing. And so this is A.1, so that's section 1 of Option A. All right, we're going to first take us to the smart board. We're going to try to get the whole section done here. All right, and what this is the understanding that the nervous system, the neural tube of an embryo, or embryonic chordates, not all embryos, is formed by the infolding of layers of tissue. All right, so thing that you want to know in animals, in vertebrate animals, those are the backbone. After fertilization, the cell is called the zygote. The zygote then divides by mitosis into a ball of cells that's called a blastula. In mammals, I believe it's called a blastocyst. The blastocyst then mitoses again and undergoes a process called gastrulation. Gastrulation, ladies and gentlemen, is going to form the 3 layers. 3 layers. These layers are called germ layers because they're not differentiated. All right, 3 layers of cells. The ectoderm, in green, they labeled here the mesoderm, and the endoderm. All right, so in chordates, all tissues, all organs are derived from those 3 layers. All right, so you can see this is meant to be read down and across because you can see the numbers here 3, 8.5. Those refers to as days of development. And one, of course, IB being IB, they're asking us to take a look at the development of an embryo of a xenopus, which is a frog. And again, up until you get about right here, day 22, until you start getting tail development, it is pretty much the same in all organisms. Okay, so let's go through some of the words in this pre-labeled diagram that we have for you. All right, so if you look at the day after day 3, you begin to see the ectoderm folding in. All right, so the region of the ectoderm folds in, starts to differentiate to form what is called the neural plate, the outermost layer, the ectoderm. The neural plate then folds inwardly, okay? You can see a few days later, folds inwardly and begins to show a groove, the neural groove, okay? And the edges, which I have on one, but it's both sides, differentiate to form a neural crest. All right, so part of the mesoderm at the base, okay, at the base there of the neural groove. See it black here. So part of the base of the mesoderm, right at the neural groove there, begins to form what is called a notochord. We'll talk about what a notochord is in just a second. So let's go over what we've done so far. You've got neural plate folding in. 4 or 5 days later, you have a neural groove flanked by 2 neural crests. You also have the mesoderm layer changing inwardly to form what is called a notochord, which you can see here. You can see, all right, and you can see the further development going in of the neural tube as well. So as you go into a day, and this is day 20 here, so I'll get a number in there, that's day 20. So that infolded groove is eventually going to close off from the rest of the neural crest and form, okay, the neural tube there that you can see prominently up on day 22. The neural tube then is going to begin to elongate in all organisms. And the neural tube is going to be responsible for the central nervous system, the brain and the spinal cord. All right, the cells of the neural crest that you can see going back and back there are going to really give you the components of the peripheral nervous system. All right, so some of the things here again. So then what did we learn in the last section? So myelin sheath, sensory neurons, motor neurons. You can go back. A couple of other things that we didn't talk about in this picture. You see the red endoderm beginning to fold in as well. And I labeled it here, but you can label it there as well into what is called the archenteron. The archenteron in a frog is going to form the digestive system of that frog, the archenteron there, the digestive cavity of the frog. And the somites on the outside in blue are going to, in a vertebrate, are going to form bones such as the rib cage. It's also going to form skeletal muscle. So you can look up and exactly see everything that comes from the ectoderm, the mesoderm, and the endoderm, other parts of. But we're really interested mostly in the nervous system here. So again, going back one more time, neural plate into neural groove, flanked by neural crest. Eventually, the neural groove closes off to form a neural tube, which is going to give you the central nervous system. Okay, the central nervous system then. All right, the mesoderm gives rise to the notochord and the endoderm into the digestive system there. And again, the notochord, I didn't give it what it's going to do. It's actually going to be part of it. It's going to give you the vertebrae discs. All right, so you see it going on. So, formation of the neural tube in a xenopus. Let's go back into the PowerPoint. We'll give you other pictures. We'll take a quick look at the notes, which will be there for you. All right, so here you see another view from IB. This is a transverse view of it all the way through. So what you see going on there, going through, again, signals. I like this picture down below better than that one up on top, but you get a different view of it. All right, and you get the actual view from an electron microscope of the, so annotating up. This is another very good picture to use, not the one we're going to use, but very good picture to use of it. There's our picture 3, 8.5, 20. All right, so this animal is a model. All right, so external fertilization. You have internal fertilization. And this is an animal model. And why do we like to use animal models? Well, because in this case, you've got external, you've got very rapid embryo development, unlike a human embryo, a little longer. All right, vital staining is available for us to see. So besides the xenopus, why do we use xenopus? We use flatworms, fruit flies, mice. Of course, always keep in mind the ethical issues of using something that is, you have to kill it to use it, to study it. All right. So the xenopus here. So again, as we discussed, I'm just going to bring up all the notes here. And this is a side panel here. All right. So this is a written description of what we were just saying. You have development from a zygote one cell to a blastula and a gastrula. So what we were just describing, neurulation, takes place during the gastrula phase. And you have your endoderm, your mesoderm, and your ectoderm. All right, so I have the innermost, the middle, and the outer, endo-inner. The archenteron, the digestive cavity in the xenopus, comes from the archenteron. All right, so we go to the ectoderm where most of the action takes place. All right, mesoderm, don't forget, mesoderm is the notochord, all right, so let's go through it again. Endoderm forms a neural plate. The neural plate folds inward for the groove. All right, so you can take a look at these and the picture there. The neural plate folds inward. All right, again, you have a neural crest flanking the sides of the neural groove. The neural plate then continues to fold inward to form the neural tube. All right, so you can see the picture of the neural tube. Again, brain and spinal cord. The neural crest, the nervous system, the neurons, the myelin sheath. All right, the somite, the vertebrae surrounding it. All right, all right, so your vertebrae, your rib cage, so everything has its little function in the nervous system. Now, again, remember the ectoderm, the mesoderm, and the endoderm also form other tissues. We still talked about the digestive system here going on. All right, so the notochord again, coming from which are your discs. All right, so that then